Okay, so I'm just at the height that you can actually see the screen above the podium, so that's good. So my name is Georgia Roberts. I'm from the U.S. National Toxicology Program, and I'm going to give you guys an overview of the NTP's research program on crumb rubber using in vitro and in vivo testing approaches. So for those of you that aren't aware, the National Toxicology Program is actually an interagency program within the United States. It is headquartered at the National Institute for Environmental Health Sciences in North Carolina, but it also consists of the National Center for Toxicological Research under the Food and Drug Administration and the National Institute of Occupational Safety and Health under the Centers for Disease Control. And this is our 40th anniversary year, and the NTP conducts research on potential human health effects that may result from environmental exposures. Okay, so I'm not going to give a lot of background, but I did want to touch on a couple points I think that are relevant to sort of framing where the NTP is approaching this. So many people may be aware that there were reports in the media of health concerns and potential health effects in athletes, particularly soccer players, soccer goalies. And the association between the exposure and the effects has been not very strong, but there are several aspects about crumb rubber as a material that I think really do warrant further investigation, which is why you're seeing so many different various groups that are interested in this material. And one of those is the potential for both widespread and long-term exposure. Within the United States, there's estimated to be over 10,000 synthetic turf fields, and this is obviously a global issue. And they're frequently and increasingly being applied in places like schools and community recreational areas and youth sports fields. My nephew, for example, started playing on synthetic turf fields when he was 4, and so you do have that potential for long-term exposure, which is obviously of concern for any material. The other thing that's interesting about this, particularly from a tox testing perspective, is that there are chemicals in tires that we know are toxic in humans, and so the question from a tox testing perspective isn't necessarily about toxicity, but more about exposure. And so we want to try and understand, all of these groups really have been focusing on exposure, and so the NTP is bringing this more from a lab setting perspective. So to help address this concern, the NTP's primary research question is under what conditions experimentally have the potential to result in systemic exposure to crumb rubber constituents. So I've broken this out into 4 arms, and I'll just walk through sort of the goals, the methods, and the results for each 4. Starting with chemical characterization of the material that we used in our studies, talk a little bit about our in vitro screening studies, and then move into the feasibility work that supported eventually our 14-day in vivo studies. So for the chemical characterization, obviously we wanted to understand the material that we were working with that would help interpret our in vitro and in vivo testing results. But we also really wanted to be able to compare the material that we use, which was a single material, to the world of crumb rubber samples that are being discussed by the other groups here. So the material that we used was received from the California Office of Environmental Health Hazard Assessment. And it was a fresh from the factory material. And so the big question for us as we move forward in interpreting our results and putting it into perspective is, how similar is our material to field samples that humans are actually being exposed to? How similar is it to new and weathered fields or indoor and outdoor fields? And so our methods that we developed, we used a variety of analytical tools to help us understand and answer that question. And so these methods were selected to sort of mirror and complement those that were being performed by other groups, so that at the end of the day, hopefully we can try and understand how relevant our studies were in terms of our crumb rubber sample versus field samples. And I'm not going to go a lot into the chemical characterization work because that's the focus of a lot of the other groups, and I would not do it justice. But I've just highlighted a couple points that I think that are relevant for when we think about the findings from our study. So we didn't see a lot of volatiles under ambient conditions. We did do higher temperature studies, but under ambient conditions, we really didn't see a lot of volatiles coming off. And in our biologic fluid simulated incubations at body temperature, we saw very few constituents that were extracted. So moving on to our in vitro evaluations, we wanted to characterize the cytotoxicity of crumb rubber or its bioaccessible constituents in multiple human cell lines. So I'll just walk through sort of what we did. So we incubated the crumb rubber in cell type specific media for varying lengths of time and temperature, up to 7 days and up to 60 degrees Celsius. And then we centrifuged the material, sterile filtered it. We didn't want to heat the crumb rubber ahead of time to sterilize it because we would have the potential to lose a lot of those volatiles that may be important, so we decided to sterile filter afterwards instead. And then we diluted this to produce what we're considering our dose response. And then cells were exposed for either 24 or 72 hours, and cytotoxicity was measured. And then we also used an LC-MS untargeted analysis approach to look at what we're calling our crumb rubber conditioned media. So we did see pretty robust cytotoxicity in human lung, skin, and small intestine cell lines. I'm showing you here just one example. These are the lung cells at 60 degrees. We did see both time and temperature dependent increases in cytotoxicity. So you can see the solid black line is the 3-hour exposure, and then as you move down, the line that's actually pretty difficult to see is the 7-day exposure. And so you see that leftward shift in dose response with cytotoxicity at lower concentrations when you increase the amount of time that the crumb rubber was incubated for. And so this response was pretty shocking, pretty striking for us. We weren't really expecting to see this much cytotoxicity. And so we wanted to do some follow-up studies in simpler fluids. And so, our initial studies were done in full media with all the serum proteins present, and we had the hypothesis that maybe there was an interaction going on with the serum proteins that might be resulting in the cytotoxicity. So our follow-up studies, we incubated the crumb rubber in phosphate buffered saline or artificial lung fluid, and then diluted that into the cell type specific media. And interestingly, we did not recapitulate that cytotoxicity. So on the right-hand side, these are, again, the lung cells. This is just at the highest temperature and a 1-day extraction. And you can see the left bank of bars is a concurrent experiment just recapitulating that cytotoxicity of the crumb rubber incubation in the media. And then you can see the PBS and ALF, there was really no large change in cell survival across the concentrations that were tested. And then to follow that up, as I said, we did an LC-MS analysis of the various medias, and using a principal component analysis, we can show that there's a chemical separation between the F12 media is circled, that's the lung media that was done with the crumb rubber in the media extraction, which was cytotoxic, that is separated from the PBS and ALF, which were actually non-toxic when tested in cells. And I don't have any names, but I will say that some of the tentatively identified compounds likely resulted from crumb rubber. Things like benzothiazole, as Randy mentioned, are pretty characteristic markers. Okay, so moving on to our in vivo studies. This is a really interesting and unique material for in vivo testing, and so before we jumped in, we wanted to just think about what would be possible to do. And so we conducted some feasibility work, and obviously we wanted to toe the line of doing tox testing, but not something that wasn't completely relevant to human exposure, and so we wanted to mimic relevant human exposure scenarios as closely as possible. So here's a video from one of our studies. Okay, that's obviously not a video from one of our studies, but we did want to mimic human exposure, but also think about just the practicality of doing in vivo testing with this material. We also needed to think about homogeneity of formulations. This is something that we usually take for granted when we're dealing with a single chemical that can be dissolved or absorbed to food. And then we needed to think about particle sizes. And so along those lines, before we started, we decided to size fractionate the material to utilize and isolate some of those smaller particles that would make it possible for us to evaluate certain routes of exposure. And so we looked at feed exposures, and so we basically just mixed the crumb rubber in feed and looked at things like whether there was visible settling or whether the formulations looked qualitatively homogeneous. We did a similar approach for mixing the crumb rubber directly into the rodent bedding. This is probably, we called this the runaround. This is probably closest to the video that I showed on the last slide. And we did similar things. We wanted to look at settling and qualitative homogeneity. In the bedding studies, we also heated the cages only up to about 31 degrees Celsius and analyzed for the release of volatiles. And then using the smallest size fraction, the 400 mesh, we wanted to evaluate the potential for gavage studies. And here we're just looking at whether or not the formulations can be dispensed to meet the gavageability requirements for an animal study. So based on that work, we decided to recombine the 3 largest size fractions, and those were used in our bedding and feed studies. And then we did deem that the 400 mesh, the smallest particle size, was acceptable for gavage studies up to 200 milligrams per mil, which is quite a bit higher than we actually ended up using based on material availability. And I'll just point you guys to the table that's on this slide. You can see that for the 400 mesh size fraction, it was less than 0.5% by weight. So there wasn't a ton of that material available, and that really drove a lot of our design considerations. So going into the 14-day studies, we wanted to, again, come back to that issue of exposure. And so we wanted to think about what routes of administration would most likely result in systemic exposure to those constituents that we might be concerned about. So based on that and based on the feasibility work, we selected female mice as our animal model. And as I've alluded to, this was really to conserve test material. So mice conserve test material compared to rats when you're talking about the gavage study because you're dosing based on body weight. But female mice conserve a lot of test material compared to male mice because for the feed and bedding studies, the animals can be co-housed. And so these are just the kind of considerations that we really had to think about in these studies that we generally might take for granted. So these 3 studies were conducted with concurrent control groups. They were all 14-day repeat dose studies. And the endpoints that we selected to look at were really, again, coming back to that exposure question. So we collected urine and plasma and analyzed that for using an untargeted analysis approach. And then we used some conventional toxicology endpoints, really with the thought that if we can detect a biological effect, that might be another tool for us to indicate that there was systemic exposure occurring. So we, thankfully, saw no impact on our conventional toxicology endpoints, which is, I think, good news for anybody that has a family member or themselves that play on these fields. And then we used an LC-MS metabolomics approach to look at the urine and plasma. Using a principal component analysis, we were not able to detect a difference between the treated and control groups for any route of exposure. And then we followed up by doing a fold change in p-value evaluation. And so in this table, I'm just showing you the number of tentatively identified compounds that had a p-value less than 0.05 and a fold change greater than 5 in the treated versus control animals. And for the purposes of today's presentation, I'll just point out that you can see for the oral gavage animals, we had a lot higher number of tentatively identified compounds for that route of exposure compared to the feed or mixed bedding. And that's not surprising when you think about those animals we were giving a direct large bolus dose to versus the more nebulous exposures that might have been occurring in the feed and mixed bedding studies. So to summarize that whirlwind tour, we did observe cytotoxicity in our in vitro studies in multiple human cell lines. But we only saw this when we did our extractions in full media with serum proteins present. When we followed up doing our extractions in PBS or artificial lung fluid, we did not see any cytotoxicity. And then using a principal component analysis, we were able to show that there was chemically a difference between the cytotoxic and non-toxic exposures. In vivo, we saw no signs of toxicity using conventional methods, and I'll just asterisk this by saying this was a very limited 14-day one-dose study. So this was not intended to necessarily evaluate toxicity, as I indicated earlier. It was really to look at that exposure question. But based on our principal component analysis mostly, there was very limited evidence of systemic exposure. So we do have posters available on our website that have the full or most of the results for these studies. That can be found easily by Googling NTP synthetic turf or NTP crumb rubber. And so if you're interested in the study results, I'd point you there and you can look at them. We'll also be publishing these results in a series of research reports, hopefully this fall. And so I'd just like to acknowledge, obviously, this is a big group effort. The chemical characterization work and 14-day studies were conducted under contract with Battelle. And then the 14-day plasma and urine analyses were done under contract with RTI. The in vitro work we did in-house in the NTP laboratory branch. And I'd be happy to take any questions. I think that will depend largely on our discussions on Thursday.